Yes, Sunil.
Yeah, Sunil is there. I'll put it here. Can you do the checking?
A little lower.
Lower. Lower? Oh yeah, I can see actually. Now this part is doing a mess up. So let's see. Okay. Yeah, but now, now, wait, I will.
Maybe
Can you see? Both I wrote both G and okay now.
Okay, this part I can now see.
yeah,
The
this works.
Oh,
left
sorry.
hand, the left hand side I cannot.
Left hand because this is coming. So what I'll do? What will I do?
You can draw a line demarcating the boundary.
No, I will also increase. How does? Oh,
Try put this one.
okay.
I think
No, put this like this.
no,
Uh,
it's
So that show everything. You don't have the clip of the.
We don't have the clipping, but that G is not appearing straight.
Keep them in the same.
okay.
Yeah, can you read both?
I can
Yeah,
read, yeah, the left hand, J I can read perfectly, Xiao like 95%. So
No,
it's
no.
actually good.
Okay.
No, it's perfect actually. Let
I
it-
think this is perfect. I can read
Yeah.
everything, right?
Yes, yes, yes.
And I have also, I think it's getting recorded as well today.
Oh, you're getting fancier by the day.
Hello.
Hello? Did it get stuck?
Let me see if I'm connected.
Yeah, I can hear you.
You can hear me, but because I'm standing in front of the iPad, but not because of my.
I see, not because of
try
the
again.
AirPods.
had a whole list of paperwork. Done.
I can't connect my headphone.
Why is this? Okay, maybe I have to change gallery. Yes.
Yeah, well, this I cannot connect. I don't know why. Maybe you have to go out to connect in the
Zoom.
no to
Oh.
so that's will be
Okay.
Bluetooth will be connected.
Okay.
There you go. Okay,
Connected.
now I'm connected.
Mithun?
Yeah, can you hear me now?
Oh yeah, I can hear you. Good.
Perfect.
You can test with Sunil as well.
And yeah, so everyone should be able to hear me, and I can hear people talking in Zoom, right? You can hear me?
Yeah,
Okay,
we can
good.
hear you. Yeah.
All right, good. All right. Now I'm going to also, I can repeat questions asked from the audience.
Sure.
Uh-huh.
Do you have any questions?
Pratim, you're supposed to sit closer today, right?
So I just want you to quickly repeat. Topo 1 was Topo 1 cuts. And
Yes, I was
without-
about to.
Oh, you are going to repeat.
I'm going to correct what I said yesterday. Yeah.
That part I would.
Yeah, it just have two different types. Yes. Uh-huh.
4 different types, but you focused on 2 yesterday, I think.
Yeah, I focus on two because two will be the one mainly for the
And
positive.
Topo 1, Topo 2, they are in bacteria and in eukaryotes all?
Yeah. Well, all those top isomers are just simply classified as 1 or 2. Depends on you count 1 strand or 2 strand. And type 1 would have the ones in the bacteria and also the ones in the eukaryotes.
And
So type 1 topoisomerase, so this is what I'm going to correct yesterday. So for topo isomerase type 1, I should say. We talked about yesterday cutting only 1 single strand.
Yes.
And I mentioned the mechanism, you don't need any ATP, right? So you just let the other strand to go around. And there are multiple types, 1A, 1B, 1C, etc. And the mechanism actually they use, I check out a bit more. Thanks for asking the question. They're actually quite different. This one is by attaching to a 5' phosphate. Those two are by attaching to a 3' phosphate. And those two are in eukaryote. And this one in bacteria. So this is the topo one we talk about in E. coli. And this one you cut and you let it through. Like I said yesterday, you can actually remove multiple supercoils at one time. For type topo 1 actually is not, it does not allow it to soar. It's more like a controlled rotation. It's about strand passing. So you only remove 1 at a time. So this actually is 1 at a time, but this one is multiple. Yeah, this is specific for bacteria.
And that is specific for eukaryotes?
For eukaryotes, yeah.
Right.
Yeah, so those are different types.
And then for topo 2, you need GPTAs.
You need ATP, not GTP.
Okay.
Yeah.
Okay. So because yesterday, I think sometimes I heard the word GTP also. What did I miss here? Because I was wondering now what is GTP. Somebody told me it's also
no, it's GTP is also energy input. So we can talk about GTP when we discuss about cytoskeletons. So a lot of cytoskeleton, like microtubule, tubulin, they hydrolyze GTP, not ATP. So
and is it because it has a different energy budget? Why do they have 2 kinds of molecules?
I don't know. Actually, that's a good question. So ATP is the most abundant source. Why some enzyme use GTP, CTP even? I don't know. I don't really know. There are quite some things. Okay. I don't know. Both all the ATP, GTP, I think their concentration are always above the saturation point.
No, I didn't understand. Could you repeat? Last sentence?
I mean, long story short, I don't know why some enzymes use ATP, some enzymes use GTP. Lots of those enzymes, actually, they have crosstalks. Like if one enzyme use ATP, people will always check whether they use GTP or not, or CTP. So they have some certain crosstalk, like they can hydrolyze all of those, but use the one most efficiently. But why some enzymes prefer one versus other, that I really do not know. Yeah, because the concentration wise, they're all really at a saturating level for all the enzymes, so it's not a binding affinity issue. I
They
don't
are all
know.
saturation, and concentration of all is also at saturation, but they might have different energy budgets. Suppose if you want a large amount of energy, you use ATP. If you have a small amount of energy,
Actually, I don't think so. The energy is stored in the triphosphate. The triphosphate groups is identical. The only thing different, even the sugar is identical. Only the base is different. So I don't think the energy is different. I think it's a binding preference, because you are recognizing the base, right? The A or T, or A or G or C, and that is different. So the binding pocket is different. But why they prefer one over I don't know.
But you said that the binding, yeah. So then the pathway could be different. It might be kinetics, right? In some chemicals, for some proteins, this one is more easily can quickly bind.
Yeah, I think the binding certainly is different, because the binding pocket have to accommodate the base. Yeah, but I actually do not know exactly why. Uh-huh.
So others can ask questions if they have.
Yeah, from yesterday, if you so people in Zoom commented that if you ask question, you speak louder, you get closer, or I'm going to repeat your questions so that people in Zoom can also hear. Uh-huh. Okay. So shall we start? Okay, thanks for bringing up the topo question, because my mind was in this eukaryotes topo as we teach the medical school students. All right, so I think yesterday I talked about supercoiling. I talked about what process inside the cell generate to supercoiling, transcription, replication, and the topo isomers are the ones to maintain the status of a supercoiling. Okay, and now we can talk a little. Maybe this is the part that Everton would really like. So supercoiling itself has quite some properties. Like if you just take a string and you trace it, it's going to buckle on itself just to minimize the energy. And that this type of structure called platooning, right? We talked about this yesterday. Like one strand just wrap around by itself, like this. This is a platooning, and this turns out if you don't have anything restricting it. If you don't have anything to restrict the movement of those plaquing on a piece of DNA, there are actually some interesting experiments. Imaging experiments have been done. So you use some intercalating dyes, and just so you can image the intensity, and you can see those plaquing actually can diffuse very rapidly on the linear DNA if you stretch the DNA. And the diffusion coefficient, I was very surprised to find out it's actually fairly high. It's like a 0.1 micrometer per second. So originally, my thoughts is that, this may be something you guys can model. It's basically just diffuse back and forth. You can actually see it on this linear DNA.
Convert this into base pairs, that would be 0.1. Okay, that's
I don't think. Okay.
1,000 base pairs per second.
So this one, there, in order to see them, they have to use a DNA like about 20 to 40 kb. So very, very long. Then you get a. So this is a huge amount of DNA. Then you can see the intensity. But if you want to compare to that in terms of distance, 0.1 microsecond per micrometer per second, it's basically like a 100 nanometers. So 10. So it's like
10 nanometers. 10 nanometers will have 10.
No, actually, it's about 600 nanometers. So you have to time of 4 if a 2D. So it's a 0.4 square root of 0.4 is about 600 nanometer per second moving.
600 nanometers. Okay,
Right,
we'll convert
direct,
that.
yeah,
We'll
yeah.
just write it down
Yeah.
somewhere.
Sorry, Jay, I missed this. So what is diffusing with this?
Later we'll convert it. Oh, it is.
So Methane was asking in Zoom what is being measured. Is that right?
Yeah, exactly.
Okay, so in this is very interesting experiment. So in this experiment, they have a double-strand DNA. Okay, so then they intercalate some sorts of a dye, so you can actually visualize the intensity of this piece of DNA. Then you can not stretch, but you tether the two ends of the DNA to an imaging surface. Now, because here you have a lots of dye intercalated, and those dye has a higher preference for negative supercoils. Okay, then you get this on your imaging plane. What you're going to see, you basically see this one long line. Then you have those high intensity spots. Then what they saw is that this spot just moving around.
I see.
Spot is just simply moving around, and they use this one. They interpret that those spots are the high intensity platinum, and the moving around of these spots is the diffusion of this platinum along the DNA. And then the measured diffusion coefficient is about 0.1 micrometer square per second.
So you're saying that in these platinum
That's both
regions,
direction.
in these platinum regions, there is enhanced diffusion compared to the other parts of the chromosome?
Well, okay, the question Methane is asking that either enhanced diffusion of those platinum region compared to the other parts of the chromosome of the DNA. I don't think this experiment can measure that, because the only visual observable is just simply this one dot fluorescent spot, and they are looking at this one fluorescent spot back and forth. And for those non-platinate DNA regions, you basically see a very diffusive fluorescence. You can't really look at the diffusion. There are probably some thermal fluctuation, but you can't really quantify it very well. So that's the only thing.
You have some size estimate of the platinum region. So if I just put it into Stokes-Einstein sort of a relation, would I get something like 0.1 micron square, or would I get something very?
So for the size of so Methane was asking the size of this platinum. I actually do not remember exactly, but it should be at least a few kb in order to have an intensity. So for example, if you have much smaller platinum, then you would not see this fluorescence spot, because it's basically by this accumulated dye, number of dye that accumulated over there, then you can see this intensity to diffuse around. So that's one measurement. Yeah.
What I'm asking is that, for example, you have some size estimate, like whatever, of some 5 nanometer of the blob that you see in your experiment, right? The intensity blob. And if I were to substitute that size into the Stokes-Einstein relationship, then I would get some sort of estimate of a diffusion coefficient. Would that be very different from what you're getting over here?
That I do not know. Okay, Methane was asking the question that you just simply estimate how much DNA you have over there. Then you use the Einstein's Stokes equation to calculate, your diffusion coefficient basically equal to your molecular size and divide by viscosity, etc. Right? So that's match up with this one. I do not know about that one. So this, I guess, first of all, this is not globular. So the Einstein's equation usually treats with cubic of the radius of your molecule. So it's globular molecule. So I don't know if this would matter. I mean, would be different or not. But I guess you could try that to calculate just how different it is. My suspicion would be actually won't be too different from each other. But although this will be a tethered, we actually should probably reduce it to one-dimensional diffusion along the DNA, maybe not to completely two-dimensional. Yeah. But this is one very interesting experiment. I think if you're going to model superconductivity, this might be something good to look at. Yes. Okay, so the question is that how far is the tether? So this tether, they're actually using lambda DNA. Lambda DNA is about 40 kilobases. 40 kilobases is actually about 10 micrometer, about 10 micrometer on the DNA, on the slice. You can actually see the whole strand. If you have this is why this type of experiment actually quite difficult. Because if you just want to like your small plasmid supercoiled, if you're not going to see, right? It's a diffraction limit. If you're within this 200 nanometer, 300 nanometer region, that you won't be able to tell the difference at all. So this is where they have to use this very long stretch DNA in order to see the diffusion along the DNA.
So when you talk about diffusion of lec2 along the DNA, here, is it that the twist is changing, or is it just that these are just waving around?
We don't know. I asked them about the same question. What they're trying to interpret is actually this whole thing is moving around. Like you just have this part unzip, this part rezip, like you're constantly moving around, and there is no energy barrier. Because I thought that you would have to pull everything off and and wrap everything, so you would have to go over energy barrier and reform it in order to move. I thought this is tremendous amount of barrier you will not be able to diffuse, but they're trying to argue actually the net energy is zero when you move this one along, and that's where they see this diffusion. I mean, I would love to see some simulation on this.
So I think I heard from collaborator of Eugene Kim, this is Decker, that
yeah, exactly, Keith Deckers. Yeah, yeah, there was.
That there is extrusion related to this diffusion. So if you switch off extrusion, does it still diffuse?
No, this is just purely. There's no other protein. This just a naked DNA. This is just a naked DNA. Yeah, that's a very different story. With the protein factor. Yeah, so I thought this is interesting. I would love to see actual some molecular dynamics simulations if this is really the case. But I was quite surprised to see that they have shown the platonic diffusion.
Pure system.
It's in vitro, totally in vitro. A purified DNA, no protein. The first add the dye to intercalate the DNA so that they become more actively supercoiled, and they put it on. Then you see this is fluorescent spots diffusing around. Yeah. Okay. So if that is really the case, then that would mean that the E. coli chromosome will be also incredibly dynamic, right? So E. coli chromosome, they have we talked about yesterday. You have a negative supercoiling density about minus 0.05, which means that every 100 turns you remove about 5 turns. So all of those negative supercoils in E. coli, based on what people have studied, that 50% of them exist in free platooning, just maybe dynamics moving around. But the other 50% of them are constrained by proteins, so they are bound by proteins, so they are not forming this type of platooning. So we can think about E. coli. So if the chromosome is like this, well, I'm not sure that you have all of those, and the negative supercoil density is only 50% exists in this type of form. The other 50% it's really more of those are being bound by many proteins called NAPs. So that's where it comes to this protein components of our nucleotide organization. Okay, so if you incorporate this type of hypothesis, then actually very early on there's some theoretical work has been done, just purely just taking this type of conformation with some plating, but then you just sometimes you add some protein at the end so that you restrain, so you do not let them diffuse too much. And just with those two, the supercoil and DNA, then you add the cell shape and also the protein concentration inside the cell. You can actually more or less compact the whole genome into the volume. So this is what we talked about yesterday. You start with this, one circle, 250 micrometer in diameter, random coil, then you reduce it to about 5 micrometer in diameter. Now you add all those inactive supercoiling and also protein concentration in the cytoplasm. Now you can recover the nucleotide and as a phase separated entity in the middle of the cell. So this is just purely by theoretical work that you probably also aware of some of those very early work that's been done.
theoretical one.
Yes, theoretical model. This is just simply calculating how many super colony you are going to have, what's the protein concentration in the cytoplasm, how those super colonies are constrained in nucleus. Then you can see all those factors come together. Then you can reduce this glob of DNA into this nuclear conformation.
But 50% is plectonin, that is experimental, right?
Yeah,
That is the data.
that's the data. That's what people suggested.
The other question is, proteins which make the cell live, can they also sit in the region of the DNA which is formed in a plectonin, or it has to first unfold the plectonin by diffusion or whatever method? Only can then proteins act or transcribe or whatever on the plectonin.
What do you mean? You mean are there any protein binding in those plectonins?
Yes, can proteins bind in
Yes, yes. So they can actually, some of those are properly stabilized by those naps. Yeah, because many of those are bent structure. DNA doesn't really like to be bent. So some of those naps, those nucleotide-associated protein actually stabilize that type of bend. So that's what I'm going to talk about next, which is the most important component of the nucleus, which is the protein. So we discuss more or less just about the DNA. Now we're going to go to this protein factor in nucleus. Yes.
Maintained by itself, or is it the macromolecular crowding that is maintaining the supercoiled form?
So the question that can supercoiled DNA maintain itself, or you need some extra factor like protein to maintain it? If you have a closed form DNA and if you have supercoiled, they are going to stay there. They are going to stay because they're just simply, you have a different topology of your DNA. They're just going to stay there. You're not cleaving the bonds or anything. They're going to exist most likely in rice or, in some opening form. They have different energy levels, but they can exist just by themselves. But then, of course, you have a protein that can help. For example, we talk about inactive supercolony, just simply opening up of the DNA. DNA doesn't like to have those single strand opened, less twisted form, but some other protein can come in and bind those regions, stabilize, so you can stay in that form for some genomic process. But if you take protein out, most likely they're going to form a plectonin or rest to stabilize conformation. Yeah, okay. So now we're going to talk about NAPs. Let me get more. NAPs are basically nucleotide-associated proteins. Many of you probably know. There are many different types of NAPs. Here, I do want to differentiate one thing of the NAPs from other DNA binding protein. There are also DNA binding protein. So other DNA binding protein, yesterday we talked about transcription factors, RNA polymerase, etc. They recognize specific DNA sequence. NAPs don't. They have some sequence specificity, but most of them, they're non-specific. You can even think of its many electrostatic interactions of those proteins. Those proteins are usually highly positively charged. They have lysine on the surface, and they just interact with the DNA non-specific. They do not look at the sequence per se. But some of them have some very interesting property. They do not recognize the sequence, but they recognize a particular DNA structure. And in E. coli, there are multiple different types. So you probably have heard of those. This IHF, this integration host factor. Then we have FACE. This called symmetric. I forgot what this one is for. The naming of FACE, and another one is HU, probably most well known. Then there is also HNS, this heat stable nucleic filament. So those are the four major types of NAPs in E. coli. Probably the most well-known one is HU. Many of you probably heard it. They are all highly abundant. So their copy number inside the cell usually is in the range of about 30 to 60,000 copies per cell. And if you convert that number to molar concentration, usually it's about 30 to 60 micromolar, right? One molecule in E. coli is about 1 nanomolar. So this is about 30 to 60 micromolar. So it's highly, highly abundant inside the cell. And what's important about those, they actually cause, they prefer to bind to DNA with different geometry. For example, one type of binding is this very, very sharp bending, and your protein is here. So this usually is caused by this integration host factor, IHF, and this angle usually is about 160 degrees. So this protein usually is really bent to the whole DNA, a very sharp turn. Now, the other naps can also cause bending, but it's much more flexible. So the binds at those, very flexibly bended DNA. This type of binding, it's very, very common. I think this can, when you have IHF at a very high concentration, other than bending DNA very, very sharply, it can bend it very flexibly. And also, this is also very typical form for HU to bind this bent.
Can I
Yes,
just
Mr.
clarify?
have a
So
question.
are you saying that these naps go and bind to these high curvature regions, or they bind and then they induce curvature?
They actually bind and induce curvature, but if your DNA is originally curved, they actually have a higher tendency to bind because you just face a confirmation that your substrate is at the right conformation you would want to already.
Okay. Okay, thank you. And this this sharp bend that you have drawn, that's like a single IHF?
Yeah, single IHF.
Okay,
yes.
So it's not necessarily clusters.
No, not a cluster. For IHF, I think people believe it's a dimer, but it's bent DNA in such a way. Yeah.
Okay.
Most of them are dimers, those are proteins. So now here I want to just mention a little for HU. So HU can bind just simply double strand DNA, just like this. They can also form higher order oligomer, but this binding affinity is really low. Usually, I think when we measure before, it's in the range of high micromolars and also totally displaceable by increasing the salt concentration, which means that this is really an electrostatic interaction. But HU has a very interesting property. If you have a DNA like kinked because you have a little nick over here, okay, or if you have a like a cruciform DNA, like you have a little kink over there, this is called the structured DNA. HU actually has a much higher binding affinity right here at this curvature. And what HU does, people think HU has a little arm and has a protein residue, can actually insert over there to stabilize this type of kink interaction. This binding is a much higher affinity compared to the regular type of binding, the linear double-strand DNA binding. So here you see that some of those proteins, they do have a preference for different DNA substrate. Okay. Now, this is another type binding. Then there is another type. This is called DNA bridging. DNA bridging. So basically, you have two DNA strands over there. Then you could have protein just binds onto it and bring two DNA segments together. This happens with HNS and also FACE. So now you can start thinking about now they can really help to compact the genome, right? So they can, in a way, when you bind to DNA, in a way you stabilize this bent conformation. We can think about you're like reducing the persistence length of your DNA. So your DNA become more flexible, right? Then they can be packed more. But then you also have this type of DNA bridging activity. You have protein bring two pieces together. Yes. I'm sorry, say it again.
So if the proteins are bound to these flexible curvatures, relatively flexible, can they induce a particular change? Can they become more sharp after the binding?
Well, this will be like a bound state for IHF. If IHF binds to your DNA, it's going to induce this sharp bending. If your DNA, I mean, this is just the final state. So this will be the state for IHF just to bind DNA and induce such a bending. And for those, they're the same. So the binding of those proteins cause this type of bending. But if your DNA is bent already like this in some of those supercoiled or open stuff, then they are also going to bind with a higher affinity, right? Yeah.
The DNA is already in a twisted form, which is freely in a twisted form, independent of the proteins.
Yes.
And the proteins are really high in the cell.
Yes.
Like the micromolar. Why don't these proteins coat that DNA?
Well,
or is there some mechanism that's keeping these proteins away from that?
That's actually a very good question. So when we think about the chromosome, it's not really just afraid those plaques just simply sit there and waiting for protein. And those protein will come and bind, right? So you have enough sign. But many of those plaques that generated actually is a very active genomic process, like transcription, replication going on. They're constant to appear and constant to disappear. So I don't think there is any known. Some of those actually protein, they can actually interact with topoisomerase to recruit topoisomerase to help with those things, to resolve those, but they are constant just generating. So when your cell is existing in a steady state, you constantly have old platinum disappeared and new platinum being generated. So it's like a binding equilibrium, right? You always have bound portion, you also have unbound portion, so existing a steady state.
And there is diffusion.
Yeah, and we will see.
IHF, which is constantly being added and removed.
Yeah, those are not static picture, right? They're constant on and off, binding, unbinding. Yeah.
So now that brings a very interesting question. I don't know what is the status of the field, but in 2009, many physicists look at the protein search problem. The protein has to find a target site, and they consider DNA to be very slowly static. And then they say, let a protein diffuse and find itself. And of course, the protein finding, the physicsy way of doing it, it's away from the experimental data by a factor of 100.
You mean faster or slower?
Slow. So if you just do as 1D diffusion and 2D diffusion.
Oh yeah, of course, that would not work.
That is but the DNA is an extremely dynamic thing. The plectoniums are constantly moving. IHF is being added, so relaxation, at least locally, it will relax. Then there is constantly loop forming and so on. So are people looking at the protein finding problem?
Well, the search problem, search problem usually it's specific to the protein you are looking at. But I think right now the general sense of many of those, for example, RNA polymers searching for a promoter or transcription factor searching for one single specific site, it's not by purely 1D diffusion, right? You're right. It's a combination of 1D and 3D together. So the protein would diffuse around. I mean, 3-dimensional diffusion inside the cytoplasm, inside the nucleus, very rapid, right? So then binds onto it, then locally 1D diffuse for a while. If it doesn't identify, then diffuse away. And that's why people say this is the most efficient way. 3D coupled with the local 1D search is the most efficient way to identify target.
Add the dynamics of the DNA. One should add that because that will also make the DNA move around a bit, which might help the protein.
In that sense, then you are changing the substrate, the binding target availability. Because then I can imagine if your target is in a plankton, might be it's harder for you to bind because you need to screen. At least you need to remove some of those constraints in order to bind.
I'm saying that the DNA conformation rather than, so my picture is like this right now. Whatever you said, 1D diffusion and 2D diffusion, the DNA is like this, and the protein is the only one which is searching. It has this suppose in here. So it is doing all sorts of 3D diffusion, 1D diffusion, 3D diffusion.
Okay, I see. So
I'm
you
saying
can
that
add
this-
the DNA diffusion as well, DNA dynamics. Okay.
Some of it has been done
Exactly.
actually.
So Mithan said that that has been done before.
I mean, some people have looked at the dynamics of the polymer chain and how it affects the search process.
Mithan said that someone has looked at the dynamics of polymer chain and how that affect the search process.
Actively, active.
No,
Well,
no, just pure fluctuations.
Just pure fluctuation, not active.
But
Yeah.
now there's a lot of activity involved. I mean, like really.
Yeah, if there are protein-protein interactions, then I can imagine that could help. Like you help recruit some protein over to bind those specific sites. I think that could happen. Yeah.
We'll
I
talk
had one
about this
question.
later after.
Okay, there is one question.
So this picture that you've drawn about IHF and this other one, that I mean, in some sense, looks like this histone picture in eukaryotes.
Yes.
Should I think about it like that, or is this something very different?
I think in general, for example, HU is actually called histone-like protein. HMS is called this heat stable nucleus histone-like protein. So people are thinking they are like histone. Histone basically you wrap the negative supercoded DNA to contain
Correct.
that, right?
Yeah.
Right. So here they actually do some of those functions. For example, HU is known to constrain negative supercoilers, but some particular mutant HU can also constrain positive supercoilers. I think actually it's a good analogy that HU function more or less like a histone, but I don't think they wrap the DNA like histone in eukaryotes.
I see.
They have different type of change of geometry, but very few. I think only one this type of wrapping would be by phase. From what I have it's like this phase would be more like histone wrapping around the DNA and form this type of a cluster polymer. But even that sense, I don't know if it's truly the same way as a histone to constrain the negative supercoils.
Okay. And this other thing I was just asking about, these DNA bridging proteins, the HNS and the FIS that you were drawing, are these like multivalent, or are their valencies known? I can see they're bivalent at least because they have to bind two parts of the DNA, but can they bind more than two?
Okay, so Mithan's question is that for those bridging proteins, what's the valency of this? See that you always only bring in two, you actually bring in multiple. So that is a little question for HMS and FACE. From what I know is that they actually only bring two pieces together, but people have report for HMS and also HU. If you have very high concentration of those proteins, you end up with, I think people call it DNA bunching. So you basically have like this, like a lattice, like all those DNA coming together. But this was really only observed in vitro at a very high concentration, so no one really knows whether they happen in vivo or not. But I suspect actually in stationary phase, yesterday we had one question about stationary. In stationary phase, there is another nucleose associated protein being highly expressed. And there people think that they're actually really packing DNA into this type of conformation. But then that is very different from this exponential phase where we need active DNA opening and packing to allow all this process to go on. And there is more or less fairly static. You have much a different transcription profile.
I see. Okay.
Okay. All right. Yes.
Will this structure be like a phase separated?
I don't know.
Like you said for CAR B, which also spreads on DNA.
Part B does that. I think more as people have shown for those proteins. I'm not sure. I'm not sure. I have to say. I mean, it's a very active research area, but I am yet to see in vitro even. For example, if you mix HU with DNA, do you see those phase condensates or not? I don't know. We haven't seen it. I mean, we also did not really look for it carefully. So I don't know. You mean for days? Oh, this is observed in vitro with very high concentration, but in vivo, it's only observed in stationary phase, but not by those protein, but different type of NAP. Very good. We have very good biologists here. Yeah. So so those are just very brief over. A lot of study have been going on for those proteins for long, but they're very promiscuous. Those proteins actually really hard to study them and know exactly, because their binding is non-specific, and even nowadays we don't even have a good crystal structure of some of those DNA and protein complex because the binding is so weak. And people have been trying to use NMR to look at their structure, but the results is just not very convincing.
Are there some numbers?
Yeah. No, you go ahead.
So are there some numbers that this is the probability of, some rate constants of attachment or whatever? So if we want to model.
Yeah.
So how do we put a certain number?
Yeah, the parameters.
Then do we say that, if a protein, one of these proteins and the DNA come in contact, the probability that they're going to get attached is low, high, so some numbers.
Yeah, okay. So there are KDs available. People have measured KD, for example, HU. The HU is the most well-studied to linear DNA and also to cruciform. Again, that number is highly variable, depends on your condition. When we measure for the cruciform, I think the KD is about in the range of about 7 or 8 micromolar, not micromolar, 7 or.
So this number is there in your paper?
Yeah, there are paper, there are KDs. Yeah, for structure specific, actually, I think it's pretty high. I think it's actually a 6 or 7 micromolar. For double strand, it's much higher KD. But we also have done single molecule tracking. We have done single molecule tracking on HU, some transcription factors, iron points. We actually have the rate constant to binds. Usually it's very transitory. So what we have shown in, at least in live cells for HU, this non-specific binding to DNA, the residence time usually is on the order of only about 5 milliseconds to 100 milliseconds. The unreached, I think it's the unreached.
That's the residence time.
The what?
Residence time.
Yeah, residence time. Yeah, the residence time we can measure. It's very, because they're very transitory. It's like constantly pushing and interacting with the DNA. So we have one paper talking about how HU help compact the genome or organize the genome. Well, we discovered that we use single molecule tracking. We track the individual HU molecules interacting with DNA. Wild type, it's very rapid. It's on and off, it's very rapid. So the residence time is really only in the range, I think, about 5 to 100 milliseconds. There are different populations. But then if we disable the non-specific binding residue of HU, those are highly charged, positively charged lysine residue, then HU become much more cytoplasmically localized. So other than just localized to the nucleus, it's actually diffused all around. And then it's basically become like a non-DNA interacting protein. But if you disable some special residue that's supposed to intercalate and for a structure specific residue, and then the diffusion behavior actually doesn't change too much compared to the wild type. It's slightly elevated. So which means that you really have a 2 type of binding mode for HU. And we also show that the non-specific binding of HU is important to agitate the chromosome to keep it in an expanded form. Because if you take that one out, the whole chromosome actually collapse, become much more condensed. And the specific binding of HU to this type of structured DNA actually is important for the condensation. So in a way that you can think about this one single small protein has 2 functions. One is to constantly keep it afloat so things can go on. But another thing, you also bind to those intermediate recombination, intermediate replication, intermediate to keep it, condensed so that you have maintaining the proper volume. So you really it's a balance of those two things together. Yeah, so I think, we even though genetics for all those proteins are very well done, we know a lot before, but actual interaction of the DNA and what's the structure, we actually still know very little. So I think maybe modeling will be able to help a lot. Okay, so those are just for the NAPs, but I know you know there are another type of proteins that many of you probably are very familiar and most interested. That's the class of protein called SMCs, the structure maintenance proteins. So they are different from the NAPs. NAPs are just so abundant, like you can think about it like a filler, just adding to DNA, shell the DNA, all those things. But SMCs are actually expressed at fairly low level. So we talk about NAPs is usually 30 to 60,000 copies per cell. Those are just a few hundred. Okay, so those are very special type of protein and very, very conserved. So they usually take a form, like you have one monomer, you have a head domain, then it goes around and coil back onto itself. So this is one monomer. So in E. coli, those are called MAC, B, E, F. So it's actually 3 proteins together. So I can get some. So this one is MACB, probably the most important one. This is also ATPase. Okay, so this one can form a dimer. Okay, so this is the dimerization domain. But underneath this one, you have 2 other proteins. One is called MAC-E, one is called MAC-F. So usually you would have is like, you have 2 MAC-F over here. I mean, I'm not drawing it correctly. And each MAC-F, you have 2 MAC-E. So in a way that this is like a monomer, this is like one enzyme of MAC-BEF. And now if you have ATP, if you have ATP hydrolysis, there are actually quite some good evidence to show this one can form a dimer. So this, of course, is a dimer, right? So, but then what you have is actually a very interesting conformation. So you still have those 2. So you actually have, actually, what they have is like this. Sorry. This one actually would kink like this. Those 2 heads are next to each other. Then you would have the MAC-F coming and joining with another MAC-F, MAC-B together. So you still have those. So they become a dimer, and people believe this dimer structure is the one that actually extruding the DNA. So the way that they extrude this type of DNA is actually very, very interesting. So I think MAC-E and MAC-B usually is about 400 copies per cell, but MAC-F is only about, yes, MAC-F is only about 200 copies. So that's why where people derived that this molecular ratio. So the binding domain of MAC-B, DNA binding domain is here. One is over here and one is right on top of the head. So what people imagine is that you would have a DNA coming in like this, then on the other side, you also have the DNA coming in over here and coming out this way. And what you can do by hydrolyzing the ATP, which is modulated by the bottom blue and yellow protein, they can constantly stream the DNA, and it has a directionality so that you end up with, you pull all of those DNA coming together, then that just keeps extruding until one moment that, oh, actually, is this the right direction? No, actually, so they actually would have DNA actually inside. Sorry, I think I draw this one in the wrong one. So if you have DNA, yeah, you have something like inside. So this loop actually become longer and longer, but then you bring those two sides of MAC-B together and it just gets stuck with each other. And that's where you would have this extruded loop, then you have this two complex right underneath. And that is many of those work regarded as really as the anchor protein for the chromosome. So we talk about all those synapses, and based on what I have read and I know, none of those can be regarded as a major anchor protein, except maybe for HU. HU has a little special role, but many of those anchor proteins are really it's denoted to the MAC-BGF protein, SMC proteins. Yes, you have questions.
Yes, I have.
Uh-huh.
So first thing is, what would be the length of these loops, average size of the loops? Is there some measurement? And along with it, okay, so that's one question. These are all related. It's related to loop forming finally.
Yeah.
And here you had another, out of those NAP proteins, there is another one which brings two strands together. I think
The HNS and the FIS.
FIS, I think.
Uh-huh.
It brings two strands together. So, I mean, to bring two strands together, they have to come close together also. Is it that the protein just diffuses around, and if there are two close strands, it gets attached?
That actually I do not know, but I don't know if MAC-BF actually has a way to bring them together.
Pardon?
I don't know if those MAC-BF will be able to help.
To come close together.
Yeah.
Oh, you're saying MAC-BF might help?
Might help to bring those DNA strands together. Then you can also have other protein at those places to help the binding. But
an average, so this is extrusion. So in bacteria, it is the MAC-BF which does the extrusion and not CTCF and so on and so forth.
Not what?
CTCF?
No. CTCF is a eukaryotic protein. It's only in eukaryotes. It's more or less a transcription factor. So it works together when it's condensing.
That's why it is. Cs talks about CTCF.
Yeah. That is a totally different protein. It's a zinc finger protein. It's different. It works when it's this type of protein. It works. It binds when the condensings in eukaryote to do the same thing, but it's different. It's totally different from this one. In the eukaryote, bacteria do not have CTCF.
Okay.
Yeah.
And I would be interested. What is the rate of extrusion? What is the average length of loops? Because the reason I'm interested in extrusion is, I think all these things like MAC-BF, and they also majorly change the conformation of the DNA in an active manner. What Nikhil is talking about is more polymerdynamics relaxation, but this is much more active.
Yeah, this is very active. It has a huge role. I will talk about MAC-BF. It's not only have a very important role in chromosome organization, compaction, but also has a very important role in segregation, in the final segregation. In terms of the rates, actually, I do not know, but David Shirat has done a little modeling. You can look into it. I think it's a molecular cell paper, 2020 or 2021. They say that just simply by the loop extrusion model, then you are going to be able to compact the whole genome. I will talk about that model, and there they have a assumed rate of it's actually very fast. So for this system, people wanted to do it in vitro so that you can really measure the rate. CTCF, you can do it, but no one can get this whole complex. Because it's huge. This arc is about 50 nanometers. It's very large. So no one has been able to get this purified complex, has biological activities, and no one has been able to do those in vitro single measure molecule measurement. Okay, so this is just
questions.
yes.
Jay, can I ask a couple of? Yeah.
Yes.
So you mentioned these are ATPases, but is it known whether the motion is active or not?
Yes, the motion, the DNA extrusion is dependent on ATP hydrolysis.
But for example,
you have to supply.
In cohesin, although it is an ATPase, I think if I understand correctly, it's mostly to do with the loading, not with the motion as such. So is the motion active, as opposed to, for example, condensin?
Okay, so let me rephrase. So your question is that the motion, which you mean is the DNA extrusion motion, is that dependent on ATP or not?
Yes.
ATP hydrolysis. Yes, it is. It is required for, because you can use ATP as a mutant, then it doesn't do the same thing. And for
but it loads onto DNA.
Condensate. Loading is a different. I don't know if loading is ATP dependent or not. In prokaryotes, the loading is done by some other protein. I'm going to talk, for example, the part B system. So there are proteins to help them to load onto the chromosome. But in eukaryotes, loading usually is done by CTCF, which has a specific DNA sequence that it recognizes. And after CTCF binds over, then the condensing can bind. There, I don't recall if the binding is ATP dependent or not, but the motion is dependent on ATP hydrolysis.
Okay. And one more thing. So, for example, in this cohesin condensin context, people, I think Decker and company now have done these experiments where they show that earlier there was this thought that this is a topological bond, in some sense, that if you have barriers which are very big, then it would not be able to cross through. But it seems like now in condensin, at least people are saying that it doesn't matter and can actually cross.
Yes. So Nathan has mentioned. Okay. Nathan mentioned a very interesting phenomena, because when we are talking about this one extruding the loops, we are talking about like this one being threaded through the loop, right? So you have to go through this little arc, and you can calculate, right? So you only have 50 nanometer, this half of the perimeter. How large your object can thread it through? And there are some beautiful experiments, like you mentioned, mentioned that you can actually tether a DNA bead like 200 nanometer inside, and it can still be extruded through. So that's basically disproof that this whole thing has to go through this arc to be threaded through. So there are different models being proposed, which that you actually don't go through the arc, you actually go from the side. I don't know exactly how it would work, but apparently you do not require the DNA to thread through this little arc. Probably it's only on the side, like going through.
So the electronics can also pass through it. So suppose, so when you're extruding, you have to really extrude the DNA backbone, or you can.
That's interesting.
DNA, bags of DNA. That's what the question is.
Okay, so Albertine asked the question, if that is the case, can you also extrude paclitanes? So, you don't have to follow the DNA backbone base after base, but you can actually jump. That
speed of the dynamics along.
Jay, can you just ask Apratim for this other person to mute? I think they have unmuted.
Okay, so someone in Zoom, could you please mute yourself if you're not asking questions, so there are some background noise. Okay, that actually I do not know. But my thoughts on this question will be depends on what's the strand extruding mechanism. Because I can think about you attaching a bead to the DNA. This scenario is different to have a paclitanen, right? Because if we just attach it to a DNA, we still have some background noise. Maybe someone can mute, please. Thank you. if you have a beads attached to a DNA, you can still follow the DNA backbone to go through. But if you have plaquetoning, you are not, unless you can actually jump around. That actually, I do not know. I do not know if that would be the case or not. My intuition is that it's actually follow the DNA backbone. Otherwise, you have to let the backbone would drop. You have to let something go, then catch the next one, which will not be consistent with the processive mechanism. Those enzymes are very, very processive. They just continuously extruding.
What is the word?
Processive. Processive basically means that you can keep doing the same motion continuously without breaking in between. So, but I don't know, but that's my thought. Okay. Now, there is another protein I think it's very important if we want to talk about chromosome organization. Other than SMS, another protein, actually one single protein called MAT-P. This protein actually was just discovered about 15 years ago. So this is a very specific protein in E. coli. This protein, if we look at the chromosome of E. coli, see this is ORI, and this is TER region. MAP-P is a DNA binding protein. It's only binds in the region of TER about 800 kb base pair region. It has like about 30 binding sites. It's a very interesting protein. MAP-P only binds over there, those sites called MAT-S. It binds over there, and there's nothing in the other part. And MAP-P itself also can form it's a dimer. Most of DNA protein binding proteins is a dimer. It has a dimerization domain over there, but it can also form a dimer of dimers. So you end up with, this DNA binding domain that you have a coil in the middle, you become a tetramer. Then this one can bind other MAT-S. Yes, can bind other. So that's where people think MAT-P actually is very important for this third domain organization. And if you delete MAT-P, this protein is not essential. Again, it's not essential. This whole third domain just spread out. It's not condensed. So that's where people think MAP-P actually is very important for the condensation. But there's still quite some controversy because for MAP-P, even if you cut off this tetramization domain, you actually don't see a huge difference. The TER is slightly mislocated, but cell doesn't appear to really care for those mechanisms. For that defect. The reason I want to bring up MAT-P is because MAT-P has some very interesting interaction with MAC-BF, protein minus, you probably know already on those studies.
Says that wherever there is MAC-P, it tries to eliminate MAC-BF.
Yes, exactly. So now I'm only introducing all those players. So I will come back about what they actually do. So now we talk about DNA, right? DNA by itself, the nuclear composition DNA, and we talk about the protein factors, the NAPS, the SMC, the MAP-P, those are the major factors. Now there's another factor we talk about, which is RNA. RNase is also one major component of nucleotide. It's only 10%. Yesterday, we talked about majority of the mRNA. Right. So the reason why mRNA is important, we talk lots of time people think it because the mRNA actually tether your nucleotide to the membrane through translation. So, we expand the chromosome. But there's another type of small RNAs. This actually is really only very new. I mean, we know there are lots of small RNAs in bacteria, so they're like 15 to 100 bases, 120 bases. But many of those, they do not have a role in the nucleotide organization. Many of them are serving like, regulators, for your RNA metabolism, etc. But this type of small RNA we talked about a little, they actually have a very special stain loop structure like this. And this RNA turned out to be also recognized by HU, by one of those NAP proteins. So if you delete just simply this one single small RNA from the nucleus just expand. So which was, it's quite interesting. I still don't understand how that would be the case. It is a highly abundant, this small RNA, but there are proposals saying that HU actually, again, you see this stem loop, it finds those kinked regions and then bring DNA together, those cruciform DNA together using those RNA. So you are like a stapler. You have two cruciform kinked DNA structure. HU bring those RNA together and then form this stem loop, then bring those two arms together. So that's as a way to condense the chromosome, to compact the chromosome. We don't know if that is the case or not. So we're actually trying to do experiment to test those type of hypotheses. But the fact is that we know if you deplete either mRNA or deplete RNA by drug treatment or genetic deletion, the nucleole is expanded. So we know that they have a role in the compaction of the nucleole. Oh, good. Oh,
Take a break or you want to continue?
Yeah, why don't we take a break? Because next, well, I'm going to talk about the methods that people look at the nuclear organization and what we know about the nuclear organization. Okay,
Keep that.
keep that. Okay, okay, okay.
You can tell them that we're taking a
Yeah.
10-minute break.
Nathan, we're taking people
Yeah.
online. We're taking a 15 minutes break. When are we coming back?
10-15 minutes.
45. So
Sure, sounds good.
3:45.
Yeah, perfect.
Okay. All right.
अभी इतना समय लगेगा ना
कौन सा ये चल रहा है?
Second washing.
4 बार करना है ना?
ये वाला कम है लास्ट में। सूट करने के लिए थोड़ा करना।
कहीं पे नहीं है जैसे कोई नहीं आता है मैंने पूरा स्टेटस was given हम लोग कर्स का देखे ना ये वाला 42 आंसर कौन सा ये भी सब्स्ट्रेट वो अच्छा जा रही है क्या वो बोला था ये हो गया अच्छा पहले तो चलो बोले ना लोग बुलाए ये क्वेश्चन तो स्क्वायर
मैं लाइट ऑफ कर रहा हूँ।
डाइल्यूट करना है क्या
तो
बोले मैंने क्वेश्चन देख दिया
सब्सक्रिप्शन के लिए कोई डायलिंग
सही बात
बड़ा साइज बड़ा होता है
हमारे साथ थोड़ा
क्या तुमने बोला
वो दिया है वो जो सीन वो जो हाँ वो ऐसे तेरे को बाबू जैसे मुझे
उसका भी एवरेज वैल्यूएशन दिया था ना तो तुम्हें
मैं करूँगा
पिछली बार नहीं
ये बहुत सारे छोटे छोटे 3 दिन करके दिया था उसने इसमें 3 3 करके दिए थे हाँ तो इसलिए बहुत सारा है इसका डबल हो गया देखो 10 है और कुछ नहीं तुम अगर छोटा छोटा प्लॉट पर हाँ इसका देखो कहाँ पे लिखा था इधर होगा बेटा ये बेटा है ये ले सकते हो मतलब अगर कम लोगे तो कम आएंगे जब तक लोगे तो छोटा ग्राफ बनेगा ना छोटा सा
फिर कंपेयर करने में इजी
एचईके का अलग से करना और एमटीडी का अलग से पर तुम्हें फिर से रिपीट तो करना ही पड़ेगा उतना हाँ बार भी बहुत कंट्रोवर्सी में आ गया था उतना सीन दिल का बहुत खराब है मैं खुद नहीं कर पा रहा कब से अब
So outings. So you're saying going into Passion temple? So I'm also going to write back to Jie Xiao and write back which is not that we should like you can go and right?
Oh, sorry, forgot. Close. All right. Okay, so let's start. So we discussed all those proteins, RNA, DNA, etc. So now how do we deduce what the chromosome is organized? So African asked me not to erase this one. That's actually a very good idea because I'm going to use this one. So there are
Can I ask
different
a
ways.
couple of things before?
Yes, Mita?
Sorry.
A question? Yes.
Yeah. So this, what is the lifetime of the loops formed by the SMCs in bacteria? So are these very long-lived, or do they have finite lifetimes?
You mean the lifetime? So Mithun was asking the lifetime of those loops. You mean the platinum?
No, the SMC loops, like the loop extrusion.
Say that again. What
the MOCBEF, the MOCBF
the
protein.
SMC lifetime?
Yeah.
What do you mean lifetime?
I mean,
You
are
mean the
these
protein's
loops,
lifetime?
no, are the loops formed by these SMC proteins, are they very stable, or do they form and dissociate?
Oh, so the question is that the loops that extruded by BEF, are they very stable or they are constantly changing? So I don't think anyone measured the loop lifetime, but people measured the MAC-BEF lifetime on the DNA. Because the MAC-BEF, when they bind onto DNA, they stay there for a while, then they would dissociate. I think that lifetime is in the range of 10 seconds. It's very 10 to 20 seconds. I don't remember actually exactly. You can look into this one paper, the early paper by David Shiraz. They did single molecule imaging of MAC-BEF. That's where they actually look at how many copy number you have. And they also measure the lifetime. If I remember correctly, I think it's in the order of 10 to 20, 30 seconds the most. So they're constantly exchanging.
Okay.
So in that sense, when they dissociate, the loop would disappear, right?
That's like a bound on
so
the
that's
lifetime.
in a way that is the loop's lifetime.
Okay.
Okay. All right, so so over the years, people become much more interested in the nuclear structure, because you can imagine this whole DNA, you cannot just simply randomly, squash them together, because you have to have all those genomic process to go on. So people start looking at what the organization of this nucleus is, and there are multiple ways to look at it experimentally. I'm an experimentalist, so I'm going to talk about those experiments. I think are going to help when you do modeling. So the first type of methods very early on is actually using genetics. I really think people are so ingeniously smart early on when they don't have all those fancy technologies. So the first type of method is by genetics. So what they did is that they use a lambda recombination. So it's basically like a bacteriophage. They basically trying to say, okay, I have a gene over here. I have another marker, reporter gene over here. What's the recombination frequency this will be? If two genes are very close to each other, you would imagine that they would have a higher combination frequency. If those two genes are far apart from each other, they would have a lower recombination frequency, right? This is because recombination also depends on the encounter of those two DNA fragments. So this was the first method they used, and people before they actually use this one to map this linear distance. Like when you say how many minutes along the chromosome, that's basically the genetic minute. But what they discovered, it's interesting that depends on where you put on the reporter on the gene, you can more or less design that there's one microdomain called ORI. What this is called ORI. Then you have some like unstructured region. Then you have another, like the left arm, the right arm, another region. Then you have another region. Like all the genes, when this one has a very high recombination rate compared to genes from this one versus the other. So in a way, you're organized into 6 different microdomain, and the genes really only recombine with genes within that one microdomain. So that was the first identification of this type of microdomains called. It's actually quite elegant work at that time. Imagine how you can actually deduce this type of organization. So for E. coli chromosome, it's something like this. But then later on, I think a couple of years ago, so this work was done by lambda recombination. So they were able to deduce this type of structure. This one was done by Frederick Bocard, yes, Frederick Bocard. That was one of the earliest work. I think it's about 15, 20 years ago. But now there is another work using called muphage. Many of you might be familiar. Muphage is used very frequently these days because it's a transposon. It just simply insert. So in a way, it's trying to assess the accessibility of your chromosome. But if you use a muphage, people show that there is no such a microdomain organization at all. Muphage basically can access everywhere. So that's where the people deduce that this chromosome actually is a well-mixed homogeneous network. We call the small world chromosome model, which this was done by Rasker. I forgot her last name, in UT Austin. It was also published just a couple years ago. But this shows that depends on your methodology, you would have fairly different views. But I can also say that because those two enzymes, the working mechanisms are very different from each other. This just simply shows that muphage, the transposon, they were able to access everywhere. So you don't really see much difference for this type of microdomain organization, right? So this is just deduced by the genetics. But now I can tell you this whole microdomain organization really gained traction because it's also validated by other methods. Another type of methods is imaging. Imaging, there are multiple ways to do imaging. So the first one is so-called called a FROST system. You probably know called fluorescent repressor operator system. So many of you probably know it's actually a very simple idea. Like you put on some DNA binding sequence over here, then you have a DNA binding protein. Now on this DNA binding protein, you put on fluorescent tag. So in a way, you can label specific segment. Now you can label, because this type of experiment is like a lifestyle experiment. Once you, not only you can measure the site, the location inside the cell, if you do like 2 over here, that using a different color, then you can measure the distance between those 2, right? So this distance, spatial distance against your linear distance can actually tell you something quite interesting, right? So if we actually did some of those work. So, but let me first finish this what people done. So because this is a lifestyle experiment, you can also measure the diffusion coefficient of those foci inside the cell. And that diffusion coefficient basically tell you how mobile your DNA fragments in those individual different domains. And what people found is that this mobility of those individual loci actually fit with this microdomain. Like if you're in this unstructured region, they're much more mobile, but if you're in the term region, it's much more restricted. So that's consistent with this microdomain idea. But what's really interesting, if you plot this D versus L, so if I plot D, which is your spatial separation in nanometers, I plot on the x-axis is L, which is your genomic distance in base pairs. So this curve actually tell you how this linear chromosome is folded in a way. So when we did some of this measurement, and some people also did this in eukaryotes, what we saw in E. coli is something like this. I think this is like about 400 nanometer. This is genomic distance. So I think goes to, I don't remember exactly, goes to about 500 kb, then it start to plateau. So we measure all those, we did those pairs, like all different positions. We saw a curve like this. And this curve, and if you are a modeler, you probably know, you can try to have different polymer models to fit this curve, and that will tell you how the chromosome is folded. And we fitted this one as a function, So as a exponent function. So my distance is proportional to the linear distance with the exponent. It's not exponential, but it's a power law relationship. Okay, so this factor turn out it's, in our measurement, it's about 0.15. So does that number ring a bell to any of you if you're doing modeling? Like if you have a long polymer, non-interacting, just collapse it. And you just simply collapse it without allowing it to reach equilibrium. Yeah, it's this fractal globule model, right? So if you just purely do that without adding anything, this should follow this power law equation, and this factor should be 1/3, like 0.33. The way that I think of it is just simply the relationship between the length and the cubic volume is third power law, right? So that's how you actually convert the linear to the volume. But ours is much smaller than that, 0.15, which suggesting that you are really compressed even more than just your polymer model would predict. And there are, of course, other models, maybe Albertine and Nathan, you all know, if you let the polymer to reach equilibrium, like you don't rapidly collapse it, you just take this polymer and just let it just randomly walk in 3-dimensional. It still follow this power law, but this factor become 0.5, become one half. So ours is clearly below that one, and which really for us, that's basically the indication that it's super compressed model, chromosome model. So that's likely is caused by supercoiling and those protein constraints, right? Okay, so this is one type of imaging. Everyone good on this?
So it starts falling off at below
I-
500 kb.
So
Where does it
below
start?
500 kb, I think it's fairly linear actually.
Up till 500 kb it's linear.
No, before 500 kb it's linear. Afterwards that plateau. Yes. It's for you collect the diameter of cell is 1 micrometer, and the diameter of the nucleode actually is smaller. We usually use about 500 to 600 nanometers because there is a certain volume. So nucleus does not expand to the whole cytoplasm. In vivo, this is live cell measurement. How do you do it in vitro? You have to isolate the nucleode out. Oh, just purely polymer. Okay. I will talk a bit, but there are some actually work was ongoing in Keith Decker's group. And you probably know that he take the whole chromosome out and strip off everything and put it back into a box. He called that chromosome in a box. And there, then you can really just study the polymer. Then you can add the protein or take out protein at different buffer conditions. Then that's where they're trying to. So that's in your sense, and that will be really just the polymer property of the DNA. But I like that system because you can really control very well, unlike inside the cell. But I suspect the inside the cell, the proteins, other factors will be much more important than just the polymers property. Yeah. Okay. So this is one type of imaging. And there's another type of imaging. It's called FISH. Probably you guys know about this one, fluorescence in situ hybridization. So what you do is that you use different oligos tie up. Each oligo has a fluorophore attached to it. Then you can do, of course, you can do this at different colors. So in a way, it is very similar to the frost system because you can have different colors, but it will be fixed cell because you have to kill the cell in order to have those oligo to hybridize. But you can do exactly the same way, measuring the distance, you just don't have the dynamics. The advantage of this method is that you can actually go for very high resolution because it's a fixed cell, and also you can apply super resolution imaging to the, so you can actually really, we can trace the whole chromosome. No one has done that in bacterial cell because it's still quite challenging. The cell is so small. But in eukaryotes, in Drosophila cells, and also in human cells, people have done this. You can actually trace one full chromosome from one end to the other, which actually is really incredible to have that resolution. You can really see the confirmation of how the chromosome is folded for using this type of method.
Did I understand it? So you're talking about tagging the entire chromosome. That's not too much, right? I mean,
Tagging the what?
entire chromosome.
Yeah, you tag the
But-
entire chromosome.
Carl begins, Paul begins, and Sukhjun, they have put in 13, 14 markers.
Yeah,
Is
but
that
that's different.
Okay.
Okay, like Paul, so Appertin was asking, do you put tags along the whole chromosome? For the frost system, you can't do that, right? You can only insert at different places. So the resolution is really low. Like even if you insert 100 or you still, 400, 4 kb, 5 kb type of resolution. But for this type of fish system, you can get a very high resolution because you can design, for example, every 1 kb, I have a tile. Every 1 kb, I have, so I can literally just walk along the chromosome and figure out how they are actually traced, right? So that gives you much higher imaging resolution, but of course it's also very tedious because the way to do it, achieve those resolution, I don't know if I will get time to talk about super resolution, is by just iteratively imaging and by clever design. But this is another way to directly visualize the conformation of the chromosome. Okay, now another way, last one is you can actually tag your proteins with some fluorescent protein. This, of course, has been used many times. When people first look at how the nucleus is distributed, the first thing they think about is just, I can tag some of those synapses, those HU, HNS, just see what their global distribution looks like. And two proteins has been really looked at. One is HU and one is HNS. Then you can do superimage imaging. You tag those with the fluorescent protein, you add them into the cell, then you see how they're distributed. But those works more or less simply just show that they're all homogeneously distributed inside the cell, cover pretty much the whole nucleus. So then people just start using them as a nucleus marker. But you cannot really figure out exactly how sequence-based those strands are arranged with each other. Early studies show HNS actually form a cluster that turn out to be an imaging artifact. So far from what we know, most of those nucleus-associated proteins, they're very uniformly distributed along the nucleus. The only thing that becomes different is these two proteins, MAT-P and also MAC-BF. Those two are different. When you add those two proteins, people found out that, let's see if I use yellow. MAC-BF really only cover this side and MAC-B cover this side. So they have very defined, so in a way they can be the label for the ORI or for the TER. Of course, but you can also use the frost system, the fluorescent tag to label where TER and ORI are. So those are different ways people can use to look at the chromosome conformation. Okay.
We've seen MACP and MUCBEF attached to different contours, different points in the contour. What was the last thing?
Yeah, not a contour. I draw it as a contour, but actually it's not. So, okay, let me just go into this part. Because if you tag a protein, you can look at their spatial distribution, right, on the chromosome. You can also look at their dynamics by using single marker tracking. So a lot of proteins that have interaction with DNA, when they bind to DNA, their diffusion coefficient usually is about 0.1 micrometer square per second. Depends on how you image, but usually within that range. That is also just the diffusion coefficient of a segment of your chromosome along the DNA. But if you are inside this whole nucleus, the diffusion coefficient usually is in the range of about 0.3 to 0.6 micrometer square per second. And if you are outside, well, let me draw this one. So if this is the nucleus, outside, usually about 1 to 2 micrometer square per second. On the DNA, it's 0.1. And if you're just diffusing around in the DNA, usually it's about 0.3 to 0.6 micrometer per second. So those are almost appear to be invariable to whatever the DNA binding protein you're looking at, MAC-BF, or of course they can be stationary, just simply stay over there, doesn't move. HU, MAC-P, or MAC-BF, they all follow more or less the same. So this type of diffusion coefficient, for example, this 0.3 to 0.6 micrometer per second diffusion, it's really just depicting how your protein is randomly, constantly interacting with your DNA. And by modulating the DNA binding affinity or by modulating the nucleus size, you can also change those diffusion coefficients. That's how you can use those numbers when you do this type of modeling. But now coming back to the MAC-BF system, this is something that's quite interesting. So let me see what's the best way to do this. Okay. So using fusion proteins. I think they did both MACB. I think this is the YFP fusion. Anyway, when they overexpress MACBEF, what they saw inside the cell that this chromosome become really like a circle, but you only have half circle, and this circle, well, this actually should be like this, only related to the origin. You don't have any MACB at the TER region. So it was figured out that MAP actually prevent in this TER region, MAP stays over there, then MACBF just cannot come over. So it's actually actively, people think it's actively stripping MACBF off. But then the contour length, it's actually quite stunning, those images, if you look at the paper. The contour length of this chromosome is, of course, much less than the full extended chromosome. And that's where the model was proposed that when MACB loads at one site, they start extruding all those DNAs until you run into another MACB dimer next nearby. Then you cannot move anymore. Then you have another. Then you continuously have this extrusion. So in a way, your chromosome looks like this. And this is how you actually compact the genome. That's how what people think that in E. coli, that's why MACB is so important in compacting the genome. And you can actually calculate, so if you're interested in the modeling, by the extrusion rate, you can actually assume extrusion rate, then also you assume a number of MACBF available in cell, and also the length and also the time scale. You can see this actually can more or less reproduce the process that they have observed under live cell imaging. So again, this is, we're going to come back to this point again, because this process turned out not only help with the compaction of the genome, but also help orient where your replication origin is, where the TER is, and also very important for the segregation. Good?
MAP-P is not essential.
MAP-P is not essential. Yes, if you have a deletion of MAP-P, they come over and occupy everything. They come back and occupy.
They're maximum around 200 MACBF.
Yeah, 400 B and F, B and E, 200 F. So it's a you can calculate, right? So, but it's not all of them are on the chromosome, right? So yeah, it's not all of them on the chromosome. So it's constantly exists in equilibrium between the binding. So
the lifetime is order of 50 to 30 seconds. So that's
yeah, it's constant. So I think actually this will be a very good model to establish. Yeah. Okay.
Other proteins would bind to this extruded DNA, or?
Well, they do actually. That's how people picture them on those extruded DNA. Because, for example, we talk about this IHF. They actually bind those very sharply bended regions. So, and you could have also HU binding some of those. So, those extruded loops are binding sites because they are distorted, bent DNA. They are actually binding site for all of those other NAPs, and people will always picture that they are all covered by other proteins. Yeah. So that is how based on imaging, you can actually get this type of organization. But I would say that's the most important. During transcription. What do you mean one part?
One part means if I compare it like petals, multiple petals. So if that region is needs to express, then only.
Oh, okay. the question is that during how such a structure will be compatible in transcription, right? So when you have transcription going on, do you only open up one region and leave the other untouched? I think that is the case. There are evidence for that. But also remember, this is a very dynamic structure. This loop is constant dissociating. I mean, the MACBF constant dissociating and forming. So I'm actually going to talk next, which is the chromosome conformation capture. That's actually going to tell how transcription is related to the whole chromosome conformation. Okay.
It's not PF essential.
Okay, so they are supposed to be essential because they are really important, but you can get deletions and cells still survive. They have tons of defect. But they can still survive. They usually have segregation defect. They usually can, some cells at 20-30% cell will be all nucleus, like they end up with no chromosome. But for bacterial cells, as long as you have a small population can revive the whole thing, they still survive. And I suspect for many of those MACB single deletion mutant, they have some suppressor mutant. No one really looked at.
I understood that in the absence of MACBF, segregation will be affected. But what did you say after that? If you don't have MACBF, then what else will happen? It will expand.
Yeah, they have to definitely
elongation growth defect.
Cell division will be delayed.
Yes.
Yes, cell division will be late. Yes, segregation is delayed. And I think the compaction of the chromosome is also changed. Yeah. So MACB really has two major roles. So one is the nucleotide organization, the compaction. Another one actually is the segregation. It helps to propel the two sister chromatin. Although people think even you don't need that one in theoretical models, it can do it yourself. Huh?
But it will slow down.
It's what?
It will slow down.
It will slow down. Yeah. Cells will slow down. Yeah, it has tremendous defect. And actually, I really think there are some secondary mutations that people did not know. Yeah, they're under the same operon. Yeah, they're all needed. Yeah, they're always like the SMC are all those three proteins together. The ATPase is the one that has this big arc. Yeah. Okay, so that brings us to the next one, which is chromosome conformation capture, which is the most popular method used right now for mapping the chromosome conformation. Well, now you can call it HiSeq because all those different methods converged. This means chromosome conformation capture. So probably many of you know what's the how this method works. What? Yeah, it's based on biochemical method. You have like this whole chromosome like all tangled together. Like you have two fragments, supposedly they're linearly apart, but they're close to each other. But by some protein, etc., then you cut them, so you end up with two pieces of DNA together. Then you ligate them, spatially separate, but they're together. I mean, linearly separate, but spatially together. Then you can ligate, and then you can sequence this whole segment. And based on the sequence reads between two DNA fragments, now you can see what's their interaction frequency is. So in a way, it's a very massively parallel. It's so much more high throughput than doing imaging, and you get the whole genomic information. So this was first put forward by Job Dacker from Nancy Claxter's lab about 20 years ago. Now it's become very, very popular because it's so high throughput. And what you usually see in this type of HiSeq data, what you see, for example, you have starting from 0 to the other end, the whole chromosome, let's say 4,000 kb. And what you usually see, you see a diagonal line, right? So the diagonal line basically means that along the chromosome, think about your chromosome. if I assume ori is here, ter is here, okay. Then this basically you're looking at the interaction along yourself. And of course, this interaction is very high, right? Because your segment nearby going to have a higher contact frequency. And this is indeed what people saw for E. coli. You basically see this one dominant axis. And there isn't anything that's interesting except for along this line, what people see, you see some type of boxes. That you have some higher density here, another density here. And because this is symmetric, so you only need to look at one axis, right? So when you see this type of boxes, this means some chromosome loci over here actually is interacting with the other side. There's some interaction over there. Okay, actually, I probably shouldn't do this because E. coli doesn't, it's some interaction over here. So which means that the chromosome is three-dimensionally folded. And when I see that this turned out to be very interesting, even though E. coli is quite boring, because this actually shows those microdomain organization as people proposed very early on using those genetic method. For example, they saw, this is the ter region that's very highly enhanced interaction in this ter region. They saw another, I think this is the ori region. Ori region, they also have a highly high interaction. Now, besides over here, they have some very low interactions down over here. And then they also see the two arms. So, they recapitulated more or less. You have this one ori microdomain, high interaction within this domain. Then you have a left, you have a right, then you have this loosely unstructured region. So those are the 6 microdomain for E. coli chromosome. So I think, with that, this microdomain hypothesis or model for E. coli, I think has been validated both by imaging, by genetics, and also by sequencing. So I think it's a fairly reasonable model to depict how E. coli's chromosome is organized. Now, if you want to look into this one more specifically, so those are just like microdomains, right? But you can also look into each domain. What's the organization? Now you can actually take this whole axis, you can try it out, and what you usually see is something like this. So this one thing people call this chromosome interaction domains, CID. It's basically the same idea as the one topological domain. That's to your question. So many of those consider, you can think about this is one domain that you have lots of this type of plankton interaction. They're very close spongy when tagged together, but you have some boundary in between to separate another domain, away from another. So those will be opened. So that's a basic idea of this type of organization. For E. coli, people think about there are 30 those chromosome interaction domains. But then the question, of course, is what to separate the domains? I don't know. There are quite some debates. So this type of structure has been seen in both E. coli, bacillus, and Chlorobacter, and people are now doing this in all other bacterial species. They saw very similar patterns, like the number of CIDs are different, and people think those are like topologically constrained domains, so that within this domain, the DNA interact with each other, but they do not propagate to the others. Where you can also think about the supercoils within this domain do not diffuse to the other. They diffuse within the domain, but do not diffuse to the other. But then the question is that what defines those boundaries? So there are two types of hypotheses of what define the boundary. One is like Albertine mentioned, it's those SMC proteins, because you can imagine two SMC complex, extruding, then they are going to run into each other, right? Because everything in between just being pushed out. Then that's basically, okay, I cannot do it anymore, I'm just going to stay there. But if that is really the case, we probably will not observe a very defined boundary because SMC is a highly dynamic back and forth. So I think it does contribute, but unlikely to define a stable boundary. And there are another hypothesis is that this boundary actually is caused by highly transcribed genes. We know that for sure in Caulobacter because there is very strong evidence for it, like those boundaries actually correlate very well with highly transcribed genes. So when you have very highly transcribed gene, what happens that we talk about, you have positive supercoiling in the front, and you have negative supercoiling at the end, right? So this is a highly transcribed gene. And those accumulated positive supercoiling and accumulated negative supercoiling, if they're not very quickly removed by top isomerase, then they would behave as a boundary. They would behave as a boundary because now everything just gets stuck over there. Nothing can come in and back because your supercoil just simply gets stuck over there. And that is a boundary. That was very well convincingly demonstrated in Caulobacter and pretty well documented in Bacillus, but not in E. coli. So E. coli is really the outback. People do think that there's some correlation, but that correlation is much lower than Caulobacter and also Bacillus. Now,
Jie,
yes.
can you just explain this part once again?
Oh,
Sorry.
okay. Hold on 1 minute, Smith. We have a question here.
This plot shows that there's no interaction between the region which is more promising.
So your question is that you only see one access line? So you don't have anything. Yes, this is actually what's odd about E. coli. So I will talk about the other species, which is very different from E. coli. E. coli is just mainly one principal axis. This is one major principal. So basically, all those interactions are fairly local along the chromosome. You have very little arm-arm interactions, unlike the other bacteria.
But when you see under a microscope and equalize the nuclei, you see it as a dense material. It's not a.
Okay, so then that's bring back. So the question is that under the microscope, the nucleus of E. coli is fairly dense, right? Okay, so there should be some interaction. So that's but why we don't see that in high C? High C actually has a spatial resolution, like how close you need to be cross-linked. And people usually take that number as about 50 nanometers. So no, 50. I think for most modular they use 50. You can adjust that number a little, but so you can think that's incredibly narrow. So even though if they appear to be very tangled with each other, they may not be captured by high C type of measurement. Okay, so Mithan, you have a question?
Yeah. Sorry, I was just asking. Can you just explain this about the supercoiling induced domain formation once, as opposed to the SMC mediated?
Okay, so Mithan asked the explanation of this not supercoiling induced domain formation about this boundary between different seeds. So
also, just the board has suddenly become very dark. I don't know.
Yeah, because we don't have any power right now.
I see. Okay. All right.
But I assume it's going to come back very soon. But I can just talk.
Sure.
So
Yeah.
Mithan was asking why transcription, why this supercoiling region define the boundary of those chromosome interaction domains. So the idea is that when you have on two sides, I don't know if you can see me or not.
I can see.
Okay, so when on the two sides you have those a lot of supercoil constrained domains over there, but in the middle part you open up, you have high transcription activity going on. Those high transcription activity would accumulate positive supercoiling on one side and negative supercoiling on the other side. And those supercoiling, if they're not removed very quickly by top isomerase, they would just simply accumulate. They would actually themselves behave as a boundary to prevent the other supercoiling to come over because energetically unfavorable. So that was the model for this type of boundary definition.
So if I I'm just trying to think. So If it was SMC mediated, generally, so you have these SMC proteins bringing together two segments and therefore at the boundary, in some sense, there is enhanced interaction because those are the
Enhanced,
enhanced contact
enhanced,
enhanced contact because the two ends are held together by the loops in that sense,
right?
which means that in these high C maps, I would see bright spots at the diagonals. In principle, if I had the resolution.
Yeah, yeah. So, yeah.
But
You will, if you have a resolution of, for example, like
in
500
principle.
base pair, then you should see those coming together.
But
Yes.
as opposed to this other picture where you have this the supercoiling induced, so that's in some sense insulation. So I would see reduced intensity at these diagonals if I were to think about it, or
So Mason is asking about, for example, if you how does that compare to MAC-BEF induced or facilitated contact?
correct.
So I can actually show you, people have done the experiment that's what I'm going to talk next. So for this type of high-C experiment, you can imagine you have a wild type organization, right? Then what you can do, you say, what's the role of HU? What's the role of FIS? What's the role of MAC-BF? You delete all those, then see what the high C map looks like, right? So that's what people have done. And it turned out, Misu, to your question, so it's fairly complicated, but I think this one group, Roman Kuzo's group, did this on E. coli. They deleted all of those different NAPs and MAC-BF. What they found out really that MAC-BF and also HU help those long-range interactions, not local, which is interesting because we think about that MAC-BF is really putting two together, but they really only help, I think it's like about greater than 300 K kilobase pairs interactions. Like if you delete MAC-BF, all those small features still remains, but it's only this larger microdomain type of interaction is diminished. And all the other NAPs like FACE, HNS, they only affect those local interactions. So in a way that they tend to think about, there are really two groups, like HU and the MAC-BF, it's really helping this global. They actually looped this whole ORI region as one big microdomain and TER as another microdomain. So MAC-BEF really only help, and HU really only help that region's organization, but not those small details, local interactions.
Okay.
So,
Thank you.
yeah. So, yes.
So for the transcription related domain, so if somebody stops the transcription process, like with antibiotics, and then what will happen?
It does change. So, but not so clearly in E. coli, but in Colobacter, you can actually show, it's actually beautiful because what they did is that they use a highly expressed one operon, they insert it at different places. And whenever you have that highly expressed operon over there, you see a clear separation. So that's where they said that the high transcription is really the one that defining the transcription and defining the boundary. But in E. coli, that was not really shown.
So, also if you have the polymerase deletion, like transient, if it's essential, will it exemplify the boundaries?
I'm pretty sure. I think you will. Although no one has done any work on a Jerry's depletion or topo 1 depletion experiment. I don't recall, or even a drug inhibition. I think it would be interesting to look at. I'm pretty sure if you inhibit Jerry's or topo 1, I think the map going to look different. Yeah. So in a way, what I'm trying to bring out over here, so we talk about all those type of nucleotide organization, etc. Those factors all play very important role, but transcription appear to be another very big factor to organize the chromosome through multiple levels, like the mRNA, RNA, protein RNA binding and also this high transcription region to define those individual domains. Okay.
Something which prevents the torsional strain from. Right?
So say that again.
This is accumulation of positive supercoil, and so there must be a block which prevents the torsional strain from going. Like, is that the reason why E. coli is you cannot get a boundary? And
E. coli, you do have the boundary,
but not as distinct as.
No, the boundary is also very distinct, but people do not know with high certainty that boundary is caused by transcription. That's the main difference. I think.
What is the cause for this holding of the DNA?
I don't know. I don't know. So it's still very active. So E. coli is really odd, but sometimes I suspect maybe E. coli is just more sensitive to supercoin. So even not highly transcribed genes will still create a boundary. So right now, actually, if you guys are interested, there is a new work come out, actually not too new anymore, 2 years old. Now they have a 500 base pair, 500 base pair high C resolution. And there, all of those interaction domains disappear because you have such a high resolution. You go into it, you don't see this overall boundary at all. While you see just really individually the gene, when you have high transcription, when you have promoter over there, now you can actually see the interactions higher actually within that region. So you actually don't have any of those. They say that CID is an artifact because you don't have enough resolution. But I don't know if that's applied to E. coli or to the others.
But also there is in high C, there is another problem that at least in E. coli, they are not synchronized, right? So different things could be doing different activity and organization. So this is some average organization.
Yeah. No, I think definitely has a level of organization. It depends on how fine you want to define it. Like if you go into, like when we talk about this mu transposon fatigue going, like everyone is accessible. But there are some global organization for this microdomain organization. I think that still exists because there are multiple evidence showing. But the only thing is that what exactly define the boundary, we don't really know. And they actually looked at all those boundaries in those HU deletion, FIS deletion, MACB deletion. They do not get affected. It's only like a 20-30% of those boundaries being affected, but majority of them stay. So that's why people don't know exactly what caused that.
So in the eukaryotes, proteins like laminin, are
yeah. Okay, very good. Yeah. So some people also think that those boundaries can be defined just simply because you have some membrane targeted operon actually being attached to the membrane. That will be similar to the case of laminin, and that could also define a boundary condition. Yeah, so that could be another reason. But you think about total, there are only about 30 CIDs based on this type of analysis. And if you look at HU deletion, MACB deletion, I think like 20, 25 of them still remain, only a couple of them disappear. So I assume it might be a combined effect of those things coming together, not a one single factor determine the boundary. Yeah.
Also more primitive than Colobacter.
What do you mean primitive?
Like primitive, it evolved earlier than. So it means that it has Secrecentus and Bacillus has more complicated proteins. That's what I mean by protein. I mean, there was E. coli and then some more chemistry or biochemistry. Proteins developed. That's what I mean by it.
Yeah, well, I don't know in the evolution. I don't know in evolution where you would place them, which one is early, which one is later. That I do not know. But E. coli is very odd. E. coli appear to be the one that just, it doesn't have many of those special factors, but more or less can do its job very well. So especially when we're going to talk about DNA segregation, you're going to see that very obviously. Yeah. So, I think the take-home message for whole E. coli organization that there are some type of microdomain organizations, and those are really just affected by MACBEF and also HU. Those two are long range, and you can more or less coarsely grained, you have this one big TER domain, and you have big this ORI domain. Then you have all of those platinum being extruded by MAC-DEF over there. Then all of those NAPS protein, the binds all over those places, except the only MAP-P binds at the chromosome, the TER region. So that's the structure. Oh, sorry, HNS actually also prefer binds to the TER region because TER region is a highly transcriptionally repressed region. So HNS, actually all of those NAPs, they are transcription factors. It suppresses or activates some gene expression. So HNS has a lot of binding sites over here. AT-rich region, they repress those horizontally transferred genes. Okay, so this is E. coli. I have a meeting at 5, but so maybe I will talk about the chromosome organization of Bacillus and Caulobacter tomorrow. That works. Okay, yeah, I probably should get ready for my meeting. Okay, that's for today. Then tomorrow I will cover the other 2 organisms, Bacillus and Caulobacter. Then I'll talk about chromosome segregation. Okay, all right. Okay, thank you.
Thanks, Ji. Thanks a lot.
Thanks so much.
Bye.
Bye-bye. Sunil looks very tired.
Not really.
I think he has had really too much of information which we have gathered over 2 years.
All right, bye.
Yes.
Bye. Bye.